Welcome, folks. Today we will be discussing metabolism and bioenergetics. And here are our learning outcomes. So we're going to talk broadly about metabolism, and we'll learn about metabolic flux. We'll need to define that and then recognize that rate-determining steps are the steps that control the flux, right? So let's zone in on that word there. We'll understand the primary ways of controlling flux through pathways, including allosteric control, covalent modification, and substrate cycles. We'll understand that high-energy compounds release a lot of energy, and the energy release is often conserved. We'll understand why various compounds are high energy, including phosphoanhydride-containing compounds, thioester-containing compounds, and redox molecules. Finally, we can use our knowledge of Gibbs free energy to understand the flow of phosphates based on free energy or the flow of electrons based on reduction potentials. Our Meet a Scientist moment today is Emmett Chappell. He is a Black man most famous on his work for bioluminescence. And this is a technology from the firefly. So fireflies light up because they have an enzyme called luciferase that works on luciferin, and it hydrolyzes ATP and releases light. And this technology, when it was discovered, has been kind of translated to modern use in labs to measure metabolism, for example, to measure levels of ATP. For example, I used a luciferase assay in my PhD project in graduate school. When we talk about metabolism, folks, we can think about it in two ways: catabolism and anabolism. In this class, we'll start off by talking about catabolism, the degradation of nutrients and harvesting energy from them. This process is generally oxidative, and it releases energy, and oftentimes that energy release is coupled to the synthesis of an energy carrier such as ATP. We will also, towards the end of this semester, touch on anabolism, so the biosynthesis of larger molecules from smaller components, and this requires an input of energy. Let's learn some vocabulary that we can use together in class. So what are metabolic pathways? What is a pathway? A pathway is a series of connected enzymatic reactions or signaling molecules. For example, this semester we're going to start off studying glycolysis, which is being shown to you here on the top portion of the area that I've circled. Metabolic pathways contain small molecules, and we'll call these reactants, substrates, intermediates, products, and all of these words we can also just substitute for metabolite. So a metabolite is a substrate or a product that's a part of a metabolic pathway. There are about 4,000 known metabolic reactions. On this slide, I'm showing you some of the known metabolic reactions in the body. The central area that I've circled, this is just glycolysis, the citric acid cycle, electron transport, and synthesis of ATP. At the end of the semester, we'll branch out a little bit beyond that. Now, we're starting with catabolism, and we'll spend the most time on that this semester. It is compartmentalized within the cell, and this is important to kind of segregate the functions. And this helps each process happen independently, while at the same time being connected in specific helpful ways. In the cytosol, we're going to learn about the process of glycolysis. In the mitochondrial matrix, we'll discuss the citric acid cycle. And in the inner mitochondrial membrane, we will discuss electron transport and oxidative phosphorylation. Catabolism, we will see make high-energy compounds of ATP, NADH, FADH2, for example. These are the major free energy sources for catabolic reactions. And the last thing that I want to kind of touch on in terms of an overview of the metabolism we're going to see for the rest of the semester is what is catabolism, a little bit in more detail, more scientifically thinking about it. We think about it as a gradual oxidation of macronutrients releasing energy. We are going to follow how glucose is oxidized from glucose, eventually, please excuse my cat, to CO2, right? Fully oxidized, most possibly oxidized. And I want us to kind of think about not just the chemist's definition of redox, gaining electrons is reduction, losing electrons is oxidation. But I want us to understand kind of more simple ways that we can look at it as biochemists, just looking at some of these organic molecules. You'll notice that reduced molecules tend to have more hydrogens, right? So for example, we're not talking about the metabolism of methane, but you'll see for glucose, it has more hydrogen. Compare that to molecules that are more oxidized. These tend to have more oxygen, right? And we will see carbon dioxide being produced this semester. So these are some things to think about as we continue to discuss metabolism.